learning how to reprogram cells in the body to make them replace the cells that need to be replaced, and giving them back to the same individual. That autologous cell therapy, I think, has broad applications in many areas.
Welcome to Unnatural Selection, a show about competition, innovation, and the strategies shaping tomorrow's frontiers. I'm Nick Encina. Each week, we speak with pioneers shaping the future.
This is episode number 14, produced in partnership with Mass General Brigham's World Medical Innovation Forum. Today, we're talking about a part of the human body most people never think about until it fails. The nervous system of the gut, sometimes called the body's second brain, controls something as basic as digestion. When it doesn't develop properly, as in Hirschsprung disease, everyday life becomes a medical crisis. In this episode, we look at a radically different approach, rebuilding what's missing instead of cutting it out. We explore how cell therapy might restore function, what it takes to do this safely in children, and why scaling biology may be one of medicine's hardest problems. Our guest is Dr. Allan Goldstein. Dr. Allan Goldstein is Surgeon-in-Chief at Mass General for Children and Chief of Pediatric Surgery at Massachusetts General Hospital, as well as the Marshall K. Bartlett Professor of Surgery at Harvard Medical School. A Yale and Harvard-trained physician, he specializes in neurointestinal diseases like Hirschsprung disease. He co-directs the Pediatric Neurogastroenterology program and leads a long-standing NIH-funded research lab focused on the enteric nervous system. Dr. Goldstein has authored over 170 peer-reviewed papers and is a recognized leader in both clinical and translational pediatric surgery. Let's go.
Allan, welcome to Unnatural Selection.
Thank you, Nick.
I always start with the same question, Allan, just to level set and give people some context about what we're going to be talking about. So in the simplest way possible, could you please let us know what field you're innovating in and what major breakthrough or improvement are you working to bring about?
Great. Well, thanks again for this opportunity to talk about my work. I'm interested in a field called neurointestinal disease. These are conditions that are caused by abnormalities in the nervous system that controls the gastrointestinal tract. The gut is fascinating in that it can function without any central nervous input. So if you take a piece of intestine and put it in a dish, if you put a pellet inside that intestine, it knows how to move that pellet from the beginning to the end of the gut. And so when the nervous system is that complicated, problems happen, and kids are either born with conditions that affect that nervous system of the gut, or they acquire it later in life. And those conditions cause a great deal of morbidity, since truly eating is one of the joys of life. And these are conditions that affect our ability to either eat or to absorb nutrients or to evacuate waste product. So those are the conditions that fascinate me, and that is the field that I focus on.
Excellent. And can you tell us what is the current standard of care for these conditions and where does it fall short?
All of these conditions, and I'll name a few in case they sort of sound familiar. There's Hirschsprung disease, which we can certainly talk about more since it's the focus of what I do, but there's also gastroparesis, where the stomach doesn't empty well, colonic dysmotility, which is associated with terrible constipation that often doesn't respond to medicines, or esophageal achalasia, where the esophagus doesn't work and people have difficulty swallowing food. All of the treatments we have now are really treating symptoms. We don't have a curative therapy. So for constipation, we give laxatives. Either they make the stool soft, or maybe they help the colon to squeeze a little better. Or for esophageal achalasia, we cut the muscle or stretch the muscle of the esophagus to let food pass more easily. So where it falls short is really in just understanding the fundamental underlying problem and treating that. If the problem is with the nerves, the treatment should be with the nerves, but we're not there yet.
So if we get to the fascinating part about this, which is your approach. So how do neurosphere-based cell therapies work and what makes them uniquely suited to this challenge?
So if we use the example of Hirschsprung disease, that's a congenital disease. Babies are born with the failure of nerve cells to develop in the lower part of the intestine. Nerve cells develop in the gut during early development. They start in the foregut, the esophagus, and they migrate all the way down to the bottom. But in 1 in 5,000 babies, they don't make it to the bottom. So that last part of the colon can be a small part, it can be the whole colon, doesn't have nerve cells, and therefore does not function, cannot evacuate waste. And right now, our only treatment is to remove that segment. But like I said earlier, if the problem is an absence of nerve cells, the treatment should be to replace the nerve cells. There's no better way to treat a disease than to actually treat the cause of the disease. So neurospheres are neuronal stem cells that grow in these in non-adherent floating conditions in the proper substrate, the proper media with growth factors. We can make neuronal stem cells form spheres of stem cells that can then be expanded, grown to large numbers, and transplanted into the part of the intestine that has no nerves. We isolate the neuronal stem cells either from the patient's own intestine, the part that's healthy, or we can also harvest neuronal stem cells from the subcutaneous fat. Both of those sources generate stem cells that we can grow into these spheres and use for cell therapy.
And if the process and if the procedure works as planned, is this a treatment or is it a cure for these diseases?
We're hoping that that'll be the cure. The fundamental problem of the disease is the absence of nerve cells. We know in mouse models with the disease that we can isolate the nerve cells, grow them, and put them back into the part of the gut that has no nerve cells, and we see a restoration of function. We see the return of motility in that segment of the gut. So in that respect, we're optimistic that cell therapy can serve as a cure. Until we do the clinical trials, though, we won't know for sure, but we're hoping that this will allow children to not need major surgery during infancy and all of the complications associated with that.
That's fascinating. One of my backgrounds is in stem cell biology. That's part of the work that I did in graduate school. And as you're talking about this, I'm hearing about all the complexity that goes into this. And so, if we think about the work that you're doing, what were some of the key technical and biological hurdles that you had to overcome to make this even theoretically possible?
Yeah, there are many challenges to this. One is we want to use autologous cells. We feel that in a baby especially, we don't want to put in allogeneic cells that would require immunosuppression. So we're going to use autologous cells from the baby's own body and therefore avoid the need for immunosuppression. The question is if where do we isolate those cells from? So we can take them from the healthy part of the intestine, but those are the nerve cells that didn't develop normally, in that child's development. So the first step was showing that actually the nerve cells that did develop are just fine. They just didn't finish their journey during embryologic development, but the ones that did make some of the journey are perfectly usable. And that took time to sort out both at the genetic, molecular, and cellular level. Next is knowing how do we grow those cells, because we really need to generate a lot of cells and they have to be of the right type. We don't know that, when we isolate these neuronal progenitor cells from the gut, we get a lot of other cells. We get fibroblasts and some immune cells. We get neurons and glial cells, and we get progenitors. So a lot of work focused on how do we expand that progenitor population, which we think is the important cell type. And that took years to sort out the growth factor conditions that do that for us. There's also the issue of cell delivery. How are we going to deliver all these cells into a segment of intestine that can be, relatively long? We've worked with large animal models to show that we can do that using endoscopy, colonoscopy, combined with ultrasound so that we can target the proper layers of the gut wall. But, so again, we actually at this point aren't exactly sure how many cells we have to deliver. How many points in the gut will we need to inject And it's hard to model that because there's no large animal with Hirschsprung disease. Mice are not a great model for this when it comes to cell delivery because their colon is tiny compared to the human colon. So again, some of it is going to boil down to good educated guesses based on preliminary data, and then just seeing how it works in the human condition.
That's excellent. And so this work is mainly in the clinical trial phase. Presumably, after this, there are going to be delivery, safety, manufacturing challenges to address as well before it gets out to patients with this disease.
Yes, absolutely. We're in the late preclinical stage. We're just finishing some final experiments, and actually, that last topic of how many cells, how many injections is a part of that work. Then you're right, the manufacturing phase comes next. That we're working with a CDRO and a cell manufacturing facility, both to show that we can do the tech transfer, and that another facility with all of the GMP, qualities is able to generate cells of the same quality, same numbers that we do in the lab and the toxicity studies that'll have to be done to make sure that there aren't any unintended consequences, that the cells don't migrate to other sites that weren't anticipated, and certainly that they don't grow out of control to form something we don't want them to. We have some good data from following these cells in mice for up to a year that show none of those consequences. So we feel confident, but those are all the steps that are going to have to happen next.
And if this therapy succeeds, how could it shift the way we think about treating not only Hirschsprung disease, but a wider range of GI disorders?
Yeah, that's what's exciting about it because we consider this neural stem cell therapy as a platform approach. The ability to isolate neural stem cells from a patient, whether it's from the gut for a gut disease or from the subcutaneous fat, which is really easily accessible, is exciting. Hirschsprung disease is a pretty rare disease at 1 in 5,000. Kids suffer, and even with a good operation, 50% of kids will have lifelong problems with GI function, fecal incontinence, severe constipation. But there are many other conditions that are more prevalent. For example, gastroparesis affects a huge number of patients. Half of patients with gastroparesis or poor gastric emptying have diabetes. We know again in preclinical rodent models that these cells can help to improve gastric emptying. Well, we're going to have to figure out who the right candidates are. Is it everybody with gastric emptying problems, or is it a subpopulation who are either missing neurons or neuronal subtypes? We're not clear yet on who the right eligibility will be. The interesting thing is, in addition to neurointestinal diseases, we've been working on a model of peripheral nerve injury. We can take out a segment of sciatic nerve, which is the major nerve that controls motor and sensory function to the leg. We can remove a segment in the mouse and replace it with a scaffold that contains these neural progenitor cells we've learned to isolate and expand. And we can show that those nerve cells are able to bridge the gap and improve function in the leg. Not get it back to normal, but at this stage, improving motor function, less muscle atrophy, better ambulatory status for the mouse. So that's really exciting. The nerve cells in the gut come from the neural crest in the embryo. So does the entire peripheral nervous system. So we think there's an opportunity here not just to treat Hirschsprung disease and not just to expand it to many other neurointestinal diseases, but also potentially to treat peripheral nerve injury, which grows the market quite a bit and shows us that we can really help a large number of patients with a variety of conditions.
That really is interesting. It's exciting. So, if I can ask you one more question, Alan, zooming way out and mainly for non-clinician audiences, um, when your technology and this platform succeeds, and it's exciting to hear that it has a broader application across the body. So, when your technology succeeds, how could this innovation changed the way that we approach human health and diseases, not just in the gut, but broader diseases? What could this mean for patients that are sick, or even in cases of prevention for people in, say, 5 or 10 years?
Yeah, that's such an interesting question. One thing that I think of as a fundamental sort of paradigm for innovation is to first not be satisfied with the status quo. The way we treat Hirschsprung disease now, removing that segment that has no nerve cells, is the same way that it was treated starting in 1948
Wow.
by Dr. Orvar Svensson, a pioneer in the disease at the time. But in nearly 80 years, we're still doing the same thing. And we need to accept the fact, and it's only been shown probably from studies in the last 10 years, that people don't do that well. We thought we were doing a great job as pediatric surgeons. And then you follow the patients longer and you realize you're not doing a great job. So I think number one is just that sort of larger view to say that we need innovation in many areas in medicine because we're not doing the best we can do for our patients. So that I would think it is the first thing. Secondly, that what I mentioned earlier is that if when we finally understand the pathophysiology of disease, we really need to target treatments that fix the underlying problem, not just address the symptoms. And so that takes, that sort of general learning from what I've been working on for me has been kind of enlightening. You'd think it's intuitive, but we're often so satisfied with the way we're treating something, we fail to realize that we haven't fixed the problem. Fixing the problem offers tremendous hope to families whose kids are born with these conditions, knowing that their child isn't going to have a big operation or take a medicine for the rest of their life. So, and then lastly, I think cell therapy generally offers a tremendous opportunity to replace cells that just don't develop, stop working normally, or do the wrong thing during life. Cell therapy is relatively new. There's a lot of excitement about iPS cells, induced pluripotent stem cells. The challenge with those is that unless they come from the person's own body, they run the risk of of rejection and the need for lifelong immunosuppression, which has lots of complications associated with it. So for now, using the body's own cells, learning how to reprogram cells in the body to make them replace the cells that need to be replaced, and giving them back to the same individual, that autologous cell therapy, I think has broad applications in many areas, many diseases, and I think it's an exciting time in the cell therapy realm.
Well, Alan, I applaud you on this tremendous development, the work that you're doing. I very much appreciate your time that you've taken in joining us, and I look forward to meeting you in person at the World Medical Innovation Forum later on this year.
Great. I look forward to seeing you there, and thank you again for this.
That was Dr. Allan Goldstein. Now, unnatural signals.
What stood out here is how innovation shows up when managing a problem is no longer enough. For decades, Hirschsprung disease has been treated by working around what's broken, removing part of the colon and hoping the system can cope. What Dr. Goldstein is doing starts from a different place. Instead of managing symptoms, he's asking what it would mean to actually restore what never formed in the first place. Choosing to use a patient's own cells isn't just a scientific detail, it's a strategic one. It removes the need for lifelong immunosuppression and clears a path towards something that could realistically scale safely in children. And while this work begins with a rare disease, it doesn't end there. The deeper signal is that rebuilding nerve function could become a platform, something that extends far beyond one condition, toward a future where biological systems can be repaired, not just managed. That shift from cutting disease out to growing function back in is the kind of change that quietly redraws what medicine believes is possible.
Thanks for listening to Unnatural Selection. If you enjoyed the episode, subscribe, leave a review, and share it with someone shaping the future. Explore more at unnaturalselection.net and connect with us on LinkedIn and Facebook. Until next time, keep evolving.